This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 150, recorded on September 23rd, 2011. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dickson Despommier.
Hey, Vince.
How you doing out there?
It's raining like hell.
It's
I'm going to start building an ark the way this weather's been lately.
I saw a cartoon at ICAC. It was a picture of two animals on shore and the ark sailing away into the distance. And one says to the other, "That was today? Darn."
Put that on your calendar.
That was really good. Also joining us from North Central Florida, where I bet the weather is great, Rich Condit.
Hi, fellas.
Hey, Rich.
How you doing?
Very well, thank you.
How's the weather, Rich?
87 degrees and, partly cloudy. It's beautiful. Nice big puffy clouds.
Nice.
I must say, however, I could use some of that rain.
Is it dry down there?
We've been in drought conditions for a long time, and, it means that the lake is down and I can't sail my boat.
Oh, dear.
So that's a bummer. But at any rate, it's good. Weather's good.
Unfortunately, Alan can't be with us today, which is too bad. Because today, Dickson, by the way,
yes,
is the 3rd anniversary of TWiV.
That's incredible, isn't it?
You and I started it on this Friday 3 years ago.
Happy birthday, fellas.
Happy birthday.
Thanks.
It happened to have been the 23rd or the 24th, but it was a Friday, this Friday.
Wow.
Did you have any expectation that 3 years from then you would be doing this?
No, I had no expectation. I figured we would do it a bit and that'd be the end of it.
Or that it would have children.
Not at all.
Right. Exactly.
So here's the story, because some listeners may not know. I had an idea to do this. I'm walking down the street one day from the parking lot. I see Dickson. I say, "I have an idea." He said, "Let's go to lunch."
Yeah, I did say that, actually.
Is that Dixon's response to anything?
Or breakfast, either one.
If we want to talk about something, he says, "Let's eat while we talk about it."
That's right.
So we went to lunch. I said, "I want to do this podcast, just you and I, just recording about viruses." He said, "Okay, let's call it This Week in Virology." I said, "Yeah, that's a good idea."
Ba-boom.
So I went to his office on this Friday.
That's right.
Brought a recorder.
Yep.
And started the first one, which is one on West Nile virus.
Yeah.
And in fact, after about an hour, my recorder got full and I didn't realize it. So I had to come back the following Friday and resume.
Yeah, and we also had an episode where Vince actually neglected to turn on the recorder, as I recall.
That too. Yeah.
We made all the mistakes.
Made a lot of mistakes, yeah. But here we are, 153 years. It's cool, and we wouldn't do it without the listeners. So thanks, everybody.
Absolutely.
You are what make us continue to go. And today we actually intend to do all email.
Oh, good.
However, yesterday 2 very important papers were published by Science magazine. And these have to do, Dickson.
Yes.
This is going to be a big surprise for you.
I think you showed them to me yesterday, but go ahead.
Oh, that's right. You were here.
I'm not old enough to have forgotten yesterday yet.
I have, and I'm younger than you are.
That's true.
These deal with XMRV and CFS, chronic fatigue syndrome. And have you had a look at these, Rich?
I have indeed.
All right. So I think we should talk about these before we get to email, don't you think, guys?
Yeah.
Oh, yeah. This is
betcha.
important.
It's a big deal. All right. So the first one is called Failure to Confirm XMRV/MLVs in the Blood of Patients with Chronic Fatigue Syndrome, a Multi-Laboratory Study. So this is by the Blood XMRV Scientific Research Working Group, which is composed of a number of laboratories. The original goal of this was to determine whether XMRV was a threat to the blood supply.
Right.
So they recruited 15 subjects previously reported to be XMRV or MLV positive. And XMRV was originally reported by Lombardi et al. In Science in 2009. And MLV positive were those reported by Low and Alter sometime later. That's why they included both patients from both studies.
These are CFS patients, which were previously reported to be positive for one or the other of these viruses.
Right. And then there were 17 healthy donors. I'm sorry, 15 healthy donors. 17 is another study I'm thinking of.
Were those healthy donors, those are people who had been tested before, is that correct?
That's right, and they were negative.
And shown negative.
That's right. So they coded the samples, blinded them.
And importantly, they also took some samples which were negative and spiked them.
Yes.
With a little bit of XMRV at an appropriate level, something matching what you might expect to serve as positive controls.
Right. Those are called spiked, right? So they blinded them, coded them, and sent them out to a number of different laboratories.
And these are fresh blood draws from all these people.
Yeah, that's right.
These are not the old samples. These
They're
are
all
fresh
new.
blood draws done by independent phlebotomists.
Right. And that's an important point, actually, which we will return to. And they sent them to good labs, right, like Abbott, CDC, FDA, NCI, WPI. And they said you can look for PCR material, you could try to culture the virus, or you could look for viral antigen. And they were given, I think they were told you could do whatever you want, whichever assays you want, basically.
Yep, whatever assays you want to use.
And that's table 1. That's the main result of the paper, which shows that no laboratory detected any PCR positive material, any culture, or any antigen, except for WPI and the Ruschetti lab at NCI.
So those are the two investigators that were on the initial science two of the investigators that were on the initial science publication that reported an association between these viruses and chronic fatigue syndrome.
Right.
And none of the other laboratories found any positives.
Right. Neither in any of the tests.
And the positives that were found by the two laboratories that did find positives were distributed between both the positive subjects and the negative subjects, the subjects that were previously found to be either positive or negative, with no statistical difference between the two.
Right. So they weren't even the same samples in both labs that were positive.
Right.
What do you think, Dickson?
I think it shows quite clearly that there's no correlation between chronic fatigue syndrome and the presence or absence of that virus.
Right.
That's what I think it shows. If I knew nothing about virology, that's what I would conclude.
Mm-hmm.
I agree.
So this group...
good science, it sounds like to me.
This group says routine blood screening for XMRV, MLV is not warranted at this time, which is their main concern. Now, they do say one thing. We cannot definitively exclude the possibility that levels of these viruses in blood may vary.
Right.
And maybe this time they were down for whatever reason.
Right.
Keeps the door open, doesn't it, Dickson?
Well, yes and no. It doesn't keep my door open, but...
so you think it's over, right?
I felt it was over for quite a while, actually.
Yeah. What about you, Rich? What's your feeling on this?
I think it's over.
Mm-hmm.
I've been reserving judgment for a long time, but there's just too much negative evidence. Put it that way. Yeah. One of the problems here is it's very difficult to prove a negative, right? Okay, true. But this is just compelling. I think that if there were really something going on here, it would have been confirmed in some other laboratories, and it simply hasn't. Right. And even where it shows up here, it shows up in a fairly in a small number of samples and in a fairly random fashion. I just think this is it.
Yep.
Well, I thought the fact that XMRV, this specific virus called XMRV, was shown to be a laboratory-generated virus.
Yes.
Really was the clincher.
Right. But not only that, but it's there's there's a good experimentation to show how one can very legitimately I mean, it's a perfectly legitimate mistake in the beginning. Okay,
Right.
very legitimately get contamination. All right, these are very difficult experiments to do. It'd be very easy for anyone to get contamination, and I don't think there's any shame in that necessarily. But once the data start coming in like this, I think you have to face the music.
There's a very good summary article at Science called False Positive, written by John Cohen and Martin Ensuring, 2 writers for Science.
Does the whole history. It's
very
really
good.
A nice
The last paragraph, Mikovits, Judy Mikovits of WPI on the original paper, says it is irresponsible to dismiss the link between XMRVs and CFS at this point. Anyone who says this is a lab contaminant has drawn the wrong conclusion and has done a disservice to the public. I'm not going to stop studying it. So
she can continue studying it if she wants, but I actually disagree with her conclusion. I think it's quite the opposite. I think at this point, it's irresponsible to keep banging on this and not at least admit the possibility, the strong possibility, the weight of the evidence arguing against it. Because,
Rich, it reminds me of a saying that we came up with in one of our TWiVs, is that we should trust science, don't trust scientists.
Right,
That's
absolutely.
Right.
So, if you have an axe to grind and you get out there with an hypothesis and you keep banging away at it and it doesn't bear fruit, those people usually don't give up.
If anything, this has raised a consciousness about chronic fatigue syndrome, certainly in my mind, and I think in a lot of other people's minds as well, as a disease that deserves some attention.
Sure.
And I'm sympathetic with those who have an issue with the fact that it has not gotten appropriate attention before, and I think that this is, it's good that it's getting attention now. But if the weight of the evidence says that there's no evidence that this virus is causing it, then to stay on this bandwagon and spend a lot of money and pursue it is misdirecting the efforts. Let's direct the efforts in an appropriate direction.
I think it's worth noting that Mikovitz's enthusiasm is probably not for XMRV, which is a very specific virus which came out of nude mice in the 90s. She's probably thinking it's a gamma retrovirus. A related virus may be causing disease. And that's possible, although we haven't seen any evidence for it yet. So if you can supply that evidence that it is a gamma retrovirus in the same family or group as XMRV, that's fine, but we haven't seen that yet.
And it's important to note that there's a funny sort of circularity to these kind of arguments, okay? If you come up to start with XMRV or some other gamma retrovirus, and that's proven to be incorrect, you can't use that as support for the notion that there is a gamma retrovirus involved.
No,
okay?
you're absolutely right on that.
It put it in your mind,
Yes,
but you're back to square one.
Yeah.
I mean, I think it's very unlikely because the original direction was from a contaminant. So the possibility that it's related to that seems awfully coincidental.
And you see this kind of thing. It's not the first time I've seen this kind of thing where somebody comes up with an argument and then they get backed into a corner and they change the argument, okay, so that it's related.
Okay? That's right. Yeah.
And yet, now the related argument really has no basis in hypothesis or anything.
Rich, Rich and Dickson, you have to know when you're wrong and say, "Yeah, I'm wrong," and move on.
Absolutely, and this is why this story is so important. Okay, because people have to realize that, as we've said so many times before, this is how science works.
Absolutely.
Okay? This article was published. That's fine. It was vetted in the public. A lot of people got on it. They tried to reproduce it. It can't be reproduced.
That's right.
Okay? And so the conclusion, the most logical conclusion, is that it's wrong. And then you have to confront that and you have to say, I was wrong, and move on. Okay?
Yeah.
It's just as important to be wrong as it is to be right. And you have to separate your ego from the problem and realize that you're wrong.
Yeah,
You're wrong
I think,
most of the time, right?
Yeah, I think that's a good point. If you can't separate it then from your ego, that's really bad.
I confront this all the time, and in fact, I find it a very interesting mental exercise. I had my most recent just the other day, where we're pursuing this hypothesis, and we got data that says the hypothesis is wrong. And every time, what goes on in my head is just fascinating. It's like I have to I have to remind myself every time that this is not about proving that I'm right. It's about finding the truth.
That's right.
And discovering that you're wrong is every bit as important and a more frequent event than discovering that you're right.
Yeah.
That's how good science is done.
That's true.
And the public has to understand that. They have to face when something is proven wrong as well. And they have to understand that that's how the process works. Okay? It's okay to be wrong. But it's not okay to insist that you're right when the weight of the evidence says you're wrong.
Yeah, but Rich, a lot of times people pick up this the cause for certainly proving that they're wrong because they're so angered at the arrogance of maintaining their position in the light of data that suggests that they are wrong. So a lot of good research does come out of these conflicts between laboratories that sort of vie for attention.
Oh, fine, yeah, that's fine with me. That's great. I mean, this has been a fascinating process.
Yeah, absolutely.
Terrific.
So there is one more study in process, and that's the Lipkin study, which is still ongoing, where he's gotten 100 positives and 100 negatives, coded them and sent them out for this same sort of study, and that'll apparently come out next year. And I want to point out that's costing us $2.3 million.
Right. Mm-hmm. And I would predict that would also be negative.
Yes.
And if it is, I hope that we put a lid on it and spend the next $2.3 million looking for something else.
Yep.
Here.
Now, there's one other paper published yesterday. It is a partial retraction. Has many of the same authors of the original paper, if not all of them. First author is Robert Silverman. It includes Lombardi, Ruschetti, Peterson, and Judy Mikovits. So they say here they have re-examined, that is, Silverman and Dasgupta have re-examined some of the samples that were used in the original study. And they have found that they are contaminated. These are preparations of peripheral blood mononuclear cells. It's DNA from them. That was originally used in the Science 2009 paper to look for XMRV DNA by PCR. They've looked at it again. They say these DNAs are contaminated with XMRV plasmid DNA.
Gee whiz.
And therefore, they are retracting Figure 1 of the original paper, which was a gel showing PCR positivity among samples from CFS patients. And then two tables in the supplementary data showing the sequences of those PCR products. So they retract essentially PCR and sequence results from that original paper. Now, this morning, Trina Suderis calls me up, and she said, Vincent, have you looked at this? I said, no, not entirely. She said, well, in the partial retraction, all of the positive CFS patient samples were contaminated with plasmid DNA. None of the negative healthy control samples were contaminated with XMRV plasmid DNA. How can that happen? And I don't know an answer for that. It's not mentioned in the paper.
And in this retraction, they do not address at all the source of the contamination. Fine.
So, Katrina said, were they blinded? And it's hard to tell in this study if they were blinded. Supposedly they were in the original 2009 study.
Actually, I'm not sure that that's true. I think one of the criticisms, I'm not sure on this, but I believe one of the criticisms of that study is that it was not properly blinded.
Well, Trina told me that, yes, it's not in the paper, but when asked at press conferences, Mikovits said they were blinded, but it was
Okay.
left out of the paper,
Okay.
which I find ridiculous because the reviewers should have insisted that it was in there. But if they were blinded, you would expect equal contamination among patient
Yes,
CFS
indeed you
samples and negatives, right?
That's right.
So this is very strange in my book. What do you think about that, Rich?
Well, I don't. I'd rather just go with the facts. They're contaminated.
But why
Fine.
are just the CFS samples contaminated?
I don't know.
Neither do I.
I don't have any data.
How about you, Dixon?
I have no guesses
Okay.
either, but
Isn't
I
it
know that
weird?
if you
Yeah,
gave a
sure,
new
it's weird.
technician 2 test tubes and asked them to label one number 1 and one number 2 and gave them 2 samples, I guarantee you that a brand new technician will mix those up.
Yeah, sure. Could be error.
Yeah,
Anyway,
error.
I'd like to see it explained because, as Rich said, there's no explanation of where the contamination occurred.
So I'm just. This may be jumping ahead a minute, but I'm just looking up on the CFID's website because I can imagine that this is going to cause a lot of stir in the CFS community, and I really have a growing respect for this CFID's organization. And there's a statement by Kimberly McCleary, the president and CEO of CFIDS, on this stuff. And I think people, patients and otherwise, who have an interest in this should read this, okay? Because it's a very rational reaction to the whole thing.
Okay.
Was that your pick of the week, Rich?
No, I got another pick of the week.
I'm actually heading out to this CFIDS association just made a scientific advisory board, and I'm on it.
Excellent.
So we're having a meeting this weekend. What timing, huh?
Yeah, great.
Excellent.
Anyway, Bob Silverman will be there.
Excellent.
I want to ask him about this this pattern of contamination. It's just weird. I don't get it. All right, so what's a partial retraction here? And I've taken out all the sequence data from this paper. What is left? Detecting virus growth in cells using antibodies, doing Western blots with sera that are not specific for XMRV, and looking for antibodies in patient sera. None of that proves that whatever is in these samples is XMRV. So can you tell me why we still have any paper left?
Right.
Exactly.
This is
Very
very
weird.
weird.
And Alan left us with a link to a blog that we have picked before for Picks of the Week called Retraction Watch that addresses that very issue, a partial retraction. And they go into their own sort of detail on it.
You think it's bizarre, right?
Yeah.
I don't know
It's a very rare event.
It's too bad we don't still have the Journal of Irreproducible Results because this could all find its way into that journal, I'm sure.
It certainly is irreproducible.
Yep.
All right,
Or
I think...
Acta retracta.
Acta retracta. I think that's a good summation of that. Probably when the Lipkin study comes out, we'll revisit this one last
Talk about
time.
it again.
Yep.
But I want to devote the rest of this TWiV to listener email. But before we do, I want to acknowledge support of this episode by Wiley Blackwell, the leading scientific publisher of books, scholarly journals, major reference works, and databases. And in September, they are offering 25% off of all microbiology and virology books. To take advantage of that, go to wiley.com/go/microbeworld. wiley.com/go/microbeworld. And there you will find a variety of books on bacteria and viruses. There's one here I thought Dickson would like, Microbial Ecology.
Love it. I love it. In fact, you can't have ecology without microbes.
Absolutely. So check that out, and we thank Wiley Blackwell for their support of This Week in Virology.
Yep.
All right, the first email is from Mary, who writes, listening to the most recent Tea Party presidential debate, I was quite upset for many reasons, but one of which was to listen to how Michelle Bachmann characterized the HPV vaccine as a dangerous government injection, with the implication that it is killing young innocent girls. I am worried already about how the movie Contagion will be received by the public as another government-created virus gone amok, and now someone else is on the national stage, implying that vaccines are dangerous and unsafe. And to hear Rick Santorum's reply about why vaccines are given was also quite scary. What to do about these kind of uninformed and politically motivated public statements?
Well, you do TWiV, I suppose. My understanding is that, although I didn't hear it, my understanding is that even Rush Limbaugh got on her case about this one.
Come on, the guy from Texas did too.
A number of people have jumped on her as this being inappropriate as it is.
What was Rick Santorum's reply?
I don't know. I didn't find that. But she has backed off a bit and said, I'm not a doctor, I'm not a scientist.
That's a fact.
Yet she still says these things.
Yeah.
Well, it's irresponsible.
It's highly irresponsible. And today the Times has an article about how long-lasting this effect will be.
Right.
To hear a politician say this could be dangerous, a lot of people believe them, and they will not have their children immunized, and she will be responsible for some deaths many, many years down the line.
Yeah, this is true. Have you now noticed that with all these political debates, at the end of the show, they have a facts checker?
Is that right? What does that person do?
Well, it's a group of people, and they're checking all of the facts from all of the debaters to make sure that what they said was actually true. And
yeah, I think that's terrific.
What did they do about this fact of hers?
Almost none of them get all the facts right. I mean, it doesn't matter which party you're for or which candidate. There are always some mistakes that they've made, and they say them in public. And so I think a politician is willing to say anything as long as nobody checks it.
God, this is so insidious. Dangerous government injection.
Yeah, it's highly insidious. And that was based on the fact that someone came up to her and said, my daughter became mentally disabled after receiving this vaccine. So based on that, Ms. Bachmann ignores all the science that's out there that's done by good, hardworking people and tells the public about this.
That's nuts.
So Rich and I heard a talk on HPV at ICAC.
Oh, yeah.
And this was a scientist reviewing the information, and there are no serious vaccine-associated side effects.
Huh.
Yeah, there's soreness at the site of injection, a little fever, lots of those. Nothing serious. There's nothing wrong with this vaccine.
And actually, your comments make me want to go back just a little bit to the discussion that we just had about XMRV and CFS, because I'm sure that a lot of the comments and correspondence that are going to go on are about how all of the results that came up negative are part of some great conspiracy, to suppress this information about CFS. And I just want to make the point that nobody involved has any motivation whatsoever to conspire against this, and it would have to be enormous conspiracy, okay?
Right.
So that kind of talk is just crazy, all right? So forget it.
So everyone has to
This is a really annoying episode. There's
is
so much in here. There's
listening
more
to the
email
United
here that's
Nations.
just.
So you have to do your thing, Mary. We do TWiV. Other people have to speak out. You have to talk to your friends, convince them. You have to do everything you can. Write. Everyone can do something different. Just keep hammering.
Good point. Like, Mary, what do you do for a living? Because obviously you see what's going on here. Talk to your friends.
Yep.
Right?
Write an article, write an op-ed piece.
You're part of the deal as well.
Yeah.
That's right.
All right, take the next one, Rich.
Trina Sideros, our buddy, writes, so I was at a birthday party for a friend's daughter yesterday, and she said to me, oh, I saw your Facebook update and clicked on This Week in Virology, and I had the podcast on while I cleaned the house. And it was really interesting. She's a mom of 2, works with kids with special needs, no prior interest in virology at all, and she cleaned the house to TWiV. Person by person, you are spreading the love of science. This little email. That's great. Thank you, Trina. I had a really good time with her at ICAC. That was really nice.
Yeah, we had a good session. If you haven't listened to 149, do so. She was really good.
And then I had to leave, and you got to visit the TWiV building.
I visited the TWiV building. It was so cool. I saw the newsroom with a whole floor of cubicles with people writing at computers,
Oh, I would have loved that.
Really quiet, and she showed me where the editors decide every day what headlines are going up, and it's cool. And it's a cool building too. It's an old Gothic building.
I was surprised that the newsroom was quiet. My impression from the movies is that it's really busy and paper flying. Of course, I guess you don't.
No, it's not like that at all.
She said that. So we were there about 3-4 p.m. and she said it gets a little noisier later. It kind of gets going in the evening.
So, towards deadlines.
But she said also there have been a lot of layoffs, so it's not
exactly
a lot of empty spaces.
Not good.
All right, the next one is from Nicola. I'm a big fan of TWiV and have been listening in for the past year or so. I thoroughly enjoyed TWiV number 141 and listening to Matt Evans talking about the new mouse model that supports hepatitis C virus entry was very exciting and an important step forward for our field. However, I wanted to make a comment on his statement that only liver hepatocytes support hep C entry to high levels, and that liver hepatocytes are the only cell type within the body that express the full complement of HCV entry receptors. Not quite true. Currently, there is a shift in the thinking that HCV is strictly a liver-tropic virus taking place within the field. I'm a postdoc working at the University of Birmingham, UK, who started working on hep C just over a year ago. I've been looking at the neurological symptoms associated with hep C in many infected individuals, and the focus of my work is to see whether these symptoms are due to the direct viral infections of brain cells. Recently, several groups, including us, reported that brain-derived neuroepithelioma cells possess all of the hep C receptors at similar levels to the hepatoma cell lines commonly used to grow hep C. Furthermore, these cells support hep C entry at levels comparable to hepatoma cells. Matt suggested that extrahepatic replication of HCV is controversial. However, this finding is not and has been validated by at least 2 hep C labs. So maybe hep C entry is not totally restricted to hepatocytes. Indeed, it suggests that there may not be a liver factor that defines hep C infection. Work on hep C is really in its infancy and has only been possible to grow the virus in culture since 2005. Every day we find out new and exciting things about this important virus, and partially thanks to Matt's work, the view that hep C is restricted to hepatocytes is being superseded. Finally, I'd like to thank you, Rich, Dick, and Alan for making virology so accessible to so many people through TWiV. The podcast also makes it really easy for people like me to keep up to date with what is going on in the whole field of virology. I do not want this email to sound like a criticism of your podcast. We at Birmingham are avid fans.
Wow.
Great.
All right, so I don't know what kind of cells these are. If they are immortalized cells, they may not be biologically relevant. They may upregulate all the receptors, so who knows? We will look forward to hearing more about this story.
Right.
Okay, Rich.
Garen writes, Hi, Twivers. I love your shows. I listen to TWiV, TWIM, TWIP, and FIB, among other science shows. Thank you for your entertainment, and a special thanks for that show over at Omega Tau. What was that?
Dickson and I did one. It was a German language podcast,
right.
Remember? With a guy over in Germany. It was about viruses, bacteria, and parasites in 90 minutes.
Where does that person find the time to listen to all these podcasts?
I don't know.
Wow.
Doesn't say what he does. At any rate,
Well, we're glad they do.
he has a pick of the week, which we're going to save for another time, and a couple of requests. I am a farmer at the moment, and I would really like a couple of shows about plant viruses and fungi.
Yeah.
Yeah, we need to do that.
Hey, he's a farmer,
Yeah.
so he listens to TWiV, TWIP, and TWIM while he's farming.
Got it.
Terrific.
All right, we'll do that.
Okay, my next request is a call for help. My wife teaches biology for a community college, and we have been searching for an acceptable video on evolution suitable for college-level students. The problem is they all stink. They all suffer from one or more of the following problems. 1, political slant. 2, religious slant. 3, no science. 4, bad science. 5, boring. 6, just a pretty animal show and tell, etc. The list of problems goes on. Can anyone please help my wife by recommending something competent? Please help. We're desperate. And unfortunately, I thought about this. I can't come up with anything. Do you guys know of anything?
I would
video.
I would recommend this person going either to the British Museum of Natural Sciences and asking them that same question, or to the American Museum of Natural History and asking them the same question. They have an educational outreach group in both of those museums. They're both intimately familiar with the nuance of evolution, and they teach to a wide spectrum of students, including grade school kids and adults. And in fact, I don't know if you've read this recently, but the American Museum of Natural History is going to start a master's program in science teacher education. So I think that those would be the best sources to answer that question.
That's a nice program. That's a great idea.
Yeah.
Good idea.
Yeah.
I thought Alan would have something here, which is it's too bad he's not here.
I might, but...
I know, but those are good suggestions, yeah.
Well, we need to do some sort of a PS on the next show with Alan in it where he can get in all his licks on this.
That's right.
Well, also, if anyone knows, any listener, we have many, many out there, let us know, and we'll pass it along. Mm-hmm. All right, the next one is really for Alan. So
Want to save it?
I'm going to save it, yeah.
Yeah.
Because I have been holding this for a while, and now he's not here. So we'll save it, and we'll move on to Jenny. Jenny writes, you could have a discussion on how simple is simple when it comes to explaining science virology to a public audience. I just finished listening to TWiV 146, where a listener was asking about a Virology for Dummies resource. And I thought it must be a bit hard for scientists, researchers themselves to dumb down their topics. That's why we don't usually have a lot of virology or immunology or science 101 type books that are more accessible to the public. I appreciate the Virology 101 episodes that you guys did previously, but I think those are still a bit technical for the layperson.
Uh-huh.
It's a tough task to accomplish, but I think there must be some golden means to getting the science right without being too engrossed in the technical details that would usually turn off the non-science person. Just my 2 cents.
Interesting.
Well, I agree with her completely. And I noticed that on this show, even though our goal is to make it presentable and understandable to a wide audience, and to some extent we do that, we do get awfully technical. And some people like that because we don't really dumb it down too much. And I agree that the Virology 101 episodes are even, get a bit technical. So, and I've been thinking a lot about this Virology for Dummies thing. We're just going to have to write the book. I know what would be in that book, and I can see the figures, all right? But I need to retire.
Yeah.
And then I need to still have energy.
You will.
You think so? You going to help me, Vincent?
I'll help you
Are we
out,
going to do this together?
Sure.
Yeah,
we'll all do it.
We'll all do it. Okay. Yeah.
But I thought I was the virus
TWiV.
dummy on this show, as a matter
Fine.
of fact.
Okay. We can.
Well, you could tell us when you can tell us when it doesn't make sense.
Virology for Dummies.
Why don't we do a TWiV Virology for Dummies series?
Here.
Like really, like maybe not for every listener, but some really basic stuff.
Okay, so it has to start with. You have to assume that you're going for an audience that is not even used to thinking about atoms or molecules, and you have to have chapters that are the molecules of life.
Yeah,
right.
Sure.
And chapters that are about cells. And chapters that are about signaling, and chapters that are about information transfer. And this can be done, all right? And then and those chapters actually can be ported to other Biology for Dummies resources as well, okay?
Yep.
Those are the basic chapters, and then you do virology on top of that. We could do this.
Yeah, we can do it as part of TWiV and then break it out as a separate podcast also for people who don't want to listen to it.
Right.
All right, we'll think about that. Thanks for stimulating that idea, Jenny. Rich, you're next.
Stan writes, hello. Forgive me, but I just recently started listening to your podcast. What? You haven't been listening to them all along? You're forgiven. You're forgiven. Forgive me, but I just recently started listening to your podcast and haven't had the opportunity to listen to all your previous episodes. I am a clinical neurologist, and I find that we are moving more and more into the realm of immune modulation for the treatment of more and more chronic diseases that are autoimmune related. Mm-hmm. The latest crew of drugs that have come out are the MAB drugs, and those are monoclonal antibodies for the uninitiated. That'll be an antibody that is highly specific for some protein. The latest crew of drugs that come out are the MAB drugs, which are humanized monoclonal antibodies. The opportunistic infection progressive multifocal leukoencephalopathy, or PML. Notice how I can just cruise right through that? Which, as you know, comes from a type of polyomavirus called the JC virus, or John Cunningham virus, is a potential problem with natalizumab treatment. I'm probably mispronouncing that. I haven't seen a lot of other opportunistic infections with using the current immunomodulators, but I was wondering if you could tell me if you know of other potential infections that we may be setting ourselves up for with the greater use of these types of drugs. It would be great if you could add a podcast about the viruses and parasites that specifically pertain to neurological disease.
Oh, that would be fun.
The presentations in neurological symptoms are particularly interesting because of many different ways that people can be affected. Syphilis and neurosyphilis are excellent examples. I love the format of your show, and you and your guests are excellent examples of what we need more of in today's media, people who actually know what they're talking about. I would suggest adding more guest appearances with practicing clinical doctors as
That's
throwing.
Good idea.
Oh, absolutely. I agree completely. As throwing in a few case presentations here and there are usually appreciated by all. I wouldn't want it to get in the way of the basic science information that your show is excellent at providing, however. So, yeah, I agree completely that there ought to be more, we ought to have more clinicians. So we actually at ICAC, there was a whole session on this problem of treating multiple sclerosis patients, which is an autoimmune disease, with these monoclonal antibodies, and that having the effect of, it must be a very specific sort of immune suppression that results in the reactivation of this polyomavirus that ordinarily just hangs out persistently in your kidneys, and it blossoms in the brain in both this circumstance and in AIDS patients. Okay. Now, I don't personally know of any other viral infections that crop up with these particular immunomodulators. Of course, immune suppression brings up all sorts of viruses, notably cytomegalovirus, other viruses that are ordinarily latent, cytomegalovirus, Epstein-Barr virus, other herpes viruses, and they make people susceptible to other things like, all kinds of stuff, adenoviruses, the polyomaviruses. There's another one that shows up in your bladder. But do you guys know of any others that show up in response to monoclonal antibodies?
I found one case report from June of this year, where this monoclonal, natalizumab, whatever.
Same one.
Associated with herpes simplex type 2 meningitis.
Gee, okay. Interesting. Another, meningitis, okay, that's meninges, so that can be...
It's not in the brain.
It's not in the brain. And that was HSV2?
Yeah.
Okay, so that's a virus that ordinarily establishes latency in neural tissue to start with.
Yeah.
So all you got to do is let that guy go, and meningitis is not an illogical result.
Yeah.
That's the only one I found. Now, at the Harvard virology retreat, I met a fellow named Igor Koralnik, and he is a neurologist who treats patients, but he also studies JC reactivation in PML. And he's very excited about TWiV. He learned about it at the retreat, and he's going to come on and do an episode with us.
Oh, great. Excellent. Oh, great.
Very nice.
Because he said he gets hundreds of emails, and he said, maybe I should make my own podcast. I said, you should do it just to answer the emails you get.
That's right. Just go to this podcast.
So he'll come on and tell us if there's anything else.
Neurology just fascinates me. I once had a neurological examination, and it was weird. It's they make you run through all of these different things, and there's such bizarre little tests, and you wonder, what are they thinking when they have me do this? Right?
Did you come out Abby normal?
No, I came out okay. I went in, I said, I know I'm just crazy, all right, but I want to make sure there's nothing really wrong. And he came out and he says, you're right, you're crazy. All right. So.
You're okay. You fit right into the rest of us.
All right. That's right.
By the way, the author of that email is an MD in Tallahassee, Florida.
Right. Notice that. So be careful what you ask for, Stan. Okay, because you may wind up in the hot seat yourself here.
Good to see clinicians listening to TWiV.
Absolutely.
I know they're busy. All right, the next one is from Jody. I'm catching up with old episodes of TWiV and was listening with interest to TWiV 103, Shots with LJ Tan. During the episode, I noted several times that there are no reported side effects from the pandemic flu vaccinations. I was wondering if you had seen this news that came out since that episode was recorded. It's a WHO statement on the association of narcolepsy with immunization with that pandemic H1N1 vaccine in Scandinavian country, Sweden and Finland, I believe. Mm-hmm. What's narcolepsy, Dickson? Do you know?
Iceland.
You have to push me awake to get me to answer that. It's the occasional falling asleep randomly in an otherwise awake person, and there's a genetic correlate to this in Dachshunds, I believe, that suffer from acute narcolepsy, and it's weird to watch the owners walk this dog breed around, and every now and then they will just collapse and fall over. Wow. And then 2 seconds later they'll rouse themselves and start walking as if nothing happened. Can you imagine suffering from narcolepsy and driving and then just suddenly going to sleep? And I think that happens more often than we think.
Yeah, this one was especially in children and adolescents, so maybe they weren't driving that much. So it was Finland and Sweden, and it was associated with the vaccine made by GlaxoSmithKline that had an adjuvant in it. Oh. Okay. Not reported anywhere else. So there's an advisory here from WHO. So she goes on, since August 2010, following widespread use of vaccines against H1N1, cases of narcolepsy, especially in children, and results have been adolescents have been reported. This has been quite big news in Finland since the first occurrences were reported last year, and almost immediately it was linked to the pandemic vaccination. As it turns out, the link is real, although the final Finnish NIH report is not due until the end of August. So actually, I do have the final report from the European Medicines Agency, and they recommend restricting use of Pandemrix, which is the name of that GSK adjuvanted vaccine. So they say there's a real statistical association of vaccination with narcolepsy. It seems to be that the vaccine is combining with the genetic makeup of the individual, because
so that these individuals have a predisposition to narcolepsy, and somehow that predisposition predisposes of this will trigger that in those individuals, apparently.
Is it transient?
And not in everybody. Well, actually, that's not addressed. A good question, Dickson, because I was wondering the same thing. I didn't see anywhere whether it was transient or not. It may be too early to tell.
Yeah.
Well,
right.
So Jody goes on, vaccination rates in Finland, as a rule, are quite high, but any risk from vaccination, real or not, has an impact on public health. This struck me as being particularly relevant given the discussion in episode 103 about vaccine safety and public perceptions. Although not on the scale of the problems from U.S. vaccinations in the 70s, he's referring to swine flu, I'm sure, and Guillain-Barré, it does dent quite badly public opinion of vaccine safety. This went through several very public rounds of confirmation and denial. I guess you caught one of the denial phases, and that's absolutely right. In 103, we said there was no association.
Right.
Although the basic description of narcolepsy sounds relatively innocuous, but as it was shown in a TV documentary here several weeks ago, it turned out many children suffered behavioral changes, some quite severe. In a few cases so bad they were literally removed from the home.
Gee.
In closing, I just want to say thanks for TWiV. I am a software engineer with no medical training, but I have a strong interest in science in general, so it is always a pleasure to listen, even if some things go right over my head. Just when I think it can't get any better, the next episode proves me wrong. Nice. So this is actually a very interesting situation, because you can never determine how a vaccine will fare when it's given to millions and millions of people.
Right.
Or any drug for that matter.
That's right.
Right.
And none of them are perfect, right?
Yeah, there's a few points here that I think are important. One is that when an association like this comes up that is proven statistically significant, the public agencies responsible for this stuff own up to it.
Right.
Okay, and they say, this looks real. Let's restrict the use of this vaccine. Let's study it further. Let's figure it out. Nobody's trying to hide anything, okay? They're taking appropriate action. And also, as you say, Vincent, there's no way that you can do a clinical trial that's big enough.
That's right.
To cover these sorts of things. So there's always a risk involved in whatever we do. You drive to the store, there's a risk, okay?
Yeah.
The benefits, they even actually point out in one of these articles that even with this, if you do an analysis of the risk-benefit, the benefits still outweigh the risks,
exactly.
Even if this is a problem. That doesn't mean that you shouldn't stop using this and use something else. By the way, the influenza vaccine that's used in the United States is not an adjuvanted vaccine. Okay, it's not this same vaccine, so this doesn't apply here. Plus, there's a genetic predisposition apparently that does this. So there's no local issue here. Nevertheless, I think it's important that people, the officials that are responsible for this, look at this and take it seriously, and they'll take appropriate steps.
Yeah.
So actually, the next writer, Anthony, wrote with an update on the same story. European Medicines Agency recommends restricting use of pandemics. What is your take on this whole thing? How could the medical industry take into account genetic mutations when designing new drugs? Is the time of one drug fits all coming to an end? Is there someone looking into the effects of mutations and how drugs behave? And more importantly, will this change the way some drugs are accepted in the U.S. without local testing if the drug has been used in another country for a long period? I recall you talking about this policy in one of your TWiVs. Unfortunately, I can't remember which one it was. Thanks for all the work you do for the program. I'm loving every episode. So right now, as we just said, you can't really take genetic makeup into consideration, but someday in the future, we'll all have our genome sequenced at birth.
You know, remember, Vincent, that's what one of those keynote talks was about at ICAC.
That's right.
Right?
Yes.
What was it?
It was about responding to Hep C drugs.
Right, because there's a certain population that responds well, a certain subpopulation that doesn't, and the investigator did an appropriate comparison, sort of a whole genome comparison. It wasn't a whole genome sequence. It was more of a single nucleotide polymorphism scan or something like that. At any rate, he identified a locus that correlates with whether or not you respond to that particular drug. So people are thinking that way, and that could be done. But, you know, no matter what, I think there's always going to be this issue that no matter what sort of clinical trials you do, there's a certain possibility that when it gets out into the population, something's going to pop up. And I think, you know, you either have to accept a certain amount of risk in this or decide you're just not going to do any of this. And frankly, I'm willing to accept a certain amount of risk.
Yeah, and another question I would raise here is that whether or not it's actually legal anywhere in the world to use children in clinical trials, and I don't think it is. So if there's a genetic predisposition and if adults have matured beyond the susceptibility stage with regards to this particular adjuvant, but children have not, then you would have missed that completely in your clinical trials, and you're totally innocent in releasing this to the general public.
The point he makes here about if you can use the results of using a drug or a vaccine in another country to get it approved in the U.S.,
yeah,
we did talk about that a while ago.
I know some drugs like that.
Can you do that, Dickson?
Well, there's one drug called Flagyl, which is otherwise known as metronidazole. And when that drug was first brought out, they were using the Ames assay to study mutagenesis, and the drug failed that test miserably, and it was banned in this country, but it was not banned in Europe. And it's used to treat trichomoniasis and other and giardia. And the drug was used in Europe for about 15 years, with complete safety, by the way, as long as you didn't drink, because there's an antabuse-like effect if you do. And the drug was then looked at again and said, it might be mutagenic in Salmonella typhimurium assays in a culture plate, but when you translate that into humans, there's no evidence that that's the case. So they allowed the drug to come in under, I guess it's compassionate use. first for the most acute cases, and then after that, of course, it's very common now to order flagyl if you've got giardiasis, even in small kids. So nobody's listening to that, and that was because of the European trials.
Yeah, I think you can use those data, but I think you have to do something in this country.
Oh yeah,
right?
yes, yeah, yes. Of course you do, but you can't just go buy a culture dish of
Sure.
bacteria to say it reverses some genetic predisposition of a bacterium.
By the way, the adjuvant in question in Pandemix is a thing called ASO3, which I saw a couple of talks on, or at least referred to at the ICAC meeting as well. Apparently, it's a very good adjuvant, and it has an interesting composition. It's got squalene, which is a fatty acid, and I
from sharks.
yeah, and I looked up. It's got squalene and DL alpha tocopherol and polysorbate 80.
That's from your gallbladder.
Tocopherol is a vitamin E derivative, has vitamin E activity, and polysorbate 80 is an emulsifier because squalene is a fatty acid, so that'll be insoluble in water. So the emulsifier makes an emulsion out of this. Yeah, I looked up a paper on this, and it's not clear. I mean, it gives a great immune response. I couldn't see anything in the paper about actual mechanism.
Yeah.
I do note that squalene is on the pathway for one of the lipid signaling molecules that we talked about before. So it makes sense to me. I'm not saying that this is what's going on, but it makes sense to me that might have immunomodulatory properties. At any rate, you can bet they're going to be having a closer look at ASO3.
Interesting that squalene should come up because just in the past week there has been a paper showing that squalamine, which is also from sharks, is antiviral.
Really?
And I'm thinking we might do that next time.
Oh, I remember the paper that we did. It was the lipid signaling was to actually dumb down the immune response, and it helped the recovery from flu, remember?
Yeah, that's right.
Right? Weird.
That's right. Okay, take the next one, Rich.
Okay. John writes, first, many thanks for the trilogy of podcasts. It's a wonderful service to humanity. But in one sense, I think you sell yourselves short. While you may entice a variety of students into various fields covered in the podcasts, you might have a more dramatic effect. The slowly evolving impact of the internet on science allows for participation by an army of interested people. It's easy to point to SETI, folding at home, or many other similar ways, and he has a wiki site of distributed computing projects, which is really terrific. Many other similar ways to contribute unused computer processing time to science, but they are not necessarily focused on your areas. I think if you mention areas that need work, you might be surprised how many people have the interest and ingenuity to help solve problems. I'm specifically thinking of cyber infrastructure, what used to be called supercomputing, where users can do computer-related projects. But there are no doubt many other tasks that can be tackled by part-time home scientists. You might therefore note that these areas and specific tasks needing to be done in the show notes. I realize that no one has extra time, except for Vincent, because he apparently purchases the unused time of others. But it's only those with the in-depth knowledge like yourselves who can identify areas that need further research. So I suggest you consider this possibility. Perhaps someday you might be considered the fathers of home microscience movement. I also applaud the comments of Alan regarding food safety and agriculture and the declining support. When the Chinese prefer U.S.-made food and drugs because they are dependable, it would be ironic, if not devastating to this country's industry, if one of our products harmed one of their citizens. Keep up the great work.
Yeah.
Less important for the podcast is the following. I am a food science PhD, known to some as Dr. Chocolate, but I now work in IT, hence some of the comments above. I work for Clemson University, where in the span of 5 years we went from not in the game to top 100 in the world in cluster technology, high performance computing. This is the sort of change that allows you to think of possibilities and not limitations. Also recognize that the rest of the world's computing power is likewise continuing to grow at a tremendous rate. We were just catching up to where we should be. Thanks again, John. Terrific.
Wow.
I found it's ironic because at the same time, I don't know where I came up with this. I know. It was a post on Google Plus. There's a recent publication in, where is it? I don't have it right with me, but at any rate, I'll get it, come back to it. A publication about how gamers using Foldit in a coordinated fashion solved an HIV protease structure.
That's right.
Okay, so you set up this Foldit thing is a game. We did this before on TWiV. I think we highlighted this. It's a game that's about protein folding, and one of the, I guess, episodes in the game or whatever was to try and solve an HIV protease structure, and a bunch of gamers got into it, and I guess their gaming efforts are combined, and they solve the structure.
Right.
Yeah. In fact, a number of people have been sending me that story for consideration in TWiV. It's just exactly what this fellow's talking about, right?
Right.
That's true.
Okay, the next one is...
It's in Nature Structural and Molecular Biology.
All right. Okay, the next one is from Peter. Hi, guys. I can't tell you how much I enjoy your show. I recommend it to all the students in my virology class, and several have commented on how much they enjoy it. I'm always behind and was listening to show number 141 this morning on my daily walk. Towards the end, someone inquired about the idea of interfering with the geometry of icosahedral capsids as a therapeutic strategy. The New Statesman article they referenced refers to the work of Ryden Twarok, a mathematician from York. Ryden and her colleague from Leeds, Peter Stockley, have actually organized a meeting called Mathematical Virology, which brings together virologists, mathematicians, and physicists to talk about mathematical principles in virus assembly. I've attended a few of them, and it is a very thought-provoking meeting. On the topic of inhibition of assembly, I wanted to point out the work of Adam Zlotnick on HBV and Mike Summers on HIV, as well as the prescient comments of Don Kaspar. Towards this end, I've attached a couple of reprints that I've authored that touch on these topics. The JMB paper cites the Zlotnick, Summers, and Kaspar papers, and the 1998 TIBTech paper lays out some of the concepts behind this approach. Perhaps you would like the references. Keep up the good work.
Nice.
So Peter is a professor of microbiology at the University of Alabama in Birmingham. And we'll post links to those stories. You go to this assembly meeting, right, in Vermont?
I have gone once, okay, and I would like to go again. I think I can't make it this time. I think there's a conflict. It was one of the best meetings I've ever been to. It was just terrific. The topic is really interesting, and it's a very very interactive meeting, a small meeting and very interactive. It's like a Gordon conference.
I'm sure they probably touch on similar subjects.
Yeah. Very good.
All right, your turn.
Stephen writes, hello, Vincent, Dickson, Alan, and Rich. I received this email regarding a new open access journal in microbiology you may be interested to know about. The forward is at the bottom. I love the show and have been an avid listener since around TWiV 70, and as many before me have done, I went back and started from the beginning of the podcast. I also thoroughly enjoyed TWIP and TWIM, especially since I can follow both from podcast one. It is simply a wonderful service the team are providing. Keep up the good work. I am from the north of Ireland, UK, and I'm currently a veterinary medicine undergraduate. I have a particular interest in immunology, microbiology, and parasites. In TWIP 6, I think it was, Dickson said, I thought, Dickson says,
We're about to duck.
Dickson said that
Yes.
horses and cows both have a rumen. This was a slip of the tongue more than anything, and isn't in any way microbiology related, but given my field of study, it made me cringe, and I felt I should say. Horses are monogastric animals and have a very small stomach for their size, due to the fact that they are continuous grazers. This is very interesting. This is also why they lack a gallbladder, as bile is continuously secreted instead of being stored. Whereas cows are ruminants, as are goats, sheep, giraffes, and deer. It is okay for horses to graze with cows, though it isn't advised to place horses with sheep. This isn't due to a possible parasite burden, but because the horse may develop a hobby for chasing the sheep. Thanks again for your for the wonderful network you provide. So you found this journal, Vincent?
I believe this is what he linked to. It's
yeah.
called Microbiology Open. It's an open access, a peer-reviewed open access journal delivering rapid decisions and fast publication of microbial science. So everyone can read these articles. So we should do some in here so that people can get them.
Right. Good idea.
All right. Does that also mean that the African grazing animals like gnus and zebras, those are all mono-stomached animals as well?
I don't know, Stephen. Stephen,
I'm going to have to ask Stephen.
I could actually find that out by just going on to the internet.
You could probably wiki them.
You could, but it would be more fun if Stephen sent us another email.
It would mean he's still listening to us and hasn't hardly rejected my statements.
I like hearing what people do. Today we've gotten a bunch of different. You can see the wide variety of people we attract.
Yep.
It's cool.
I think the horse digestive system is really interesting as well. They move stuff through so fast, right, that they don't even need a gallbladder.
That's cool.
Because they got a very small stomach. Pretty
Well, I do know that half of what they excrete is undigested food.
Right.
So that actually serves as a fertilizer. That's the way the buffalo do it also. But I think there are ruminants. I think buffalo are ruminants.
And they got to eat all the time.
All the time.
Right.
Yep.
Okay.
All right. The next one is from John. I was listening to TWiV 144 and felt like I learned a lot from it, particularly the zinc finger DNA paper. I had to rewind and listen a few times and probably still missed a lot since the paper and discussion dealt with some pretty in-depth molecular biology. This led me to a few questions. Yeah, that was a pretty intense episode.
It was pretty intense, but really great.
It's good that he stuck with it.
Yeah.
Hey, since they can make this zinc finger thing that will basically clobber one particular gene and even encode it in a virus's genetic material, I wonder whether they have looked at using this to remove or inactivate the HIV provirus from infected cells. Couldn't you use this trick to make a virus that would infect T cells and inactivate the HIV lurking in their DNA? If I understand correctly, that HIV is usually latent in T cells till they're activated to fight an infection. This might be useful for massively decreasing the number of T cells silently carrying the virus. It seems like this could be done to stem cells before transplantation. Didn't one of your earlier guests talk about doing this to treat some kinds of blood cancers using an oncolytic virus? But maybe it could also be done as part of antiretroviral therapy to decrease the number of places the virus might be hiding out and make it less likely that the virus will eventually mutate to become resistant to the drugs. So he's asking whether you could knock out the proviral DNA with a zinc finger, right?
Yeah. First of all, he's obviously completely tuned in.
Right.
The questions are right on the money. Okay. And we did an episode previously about using what was it? It's like a Cree nuclease. They called it the tree nuclease.
Tree, yeah, that's right.
Engineered nuclease. So it's a similar kind of thing. It's a different enzyme, but a similar type of thing that would recognize flanking sequences on an HIV protease and clip it out. So people are thinking about this. They're doing experiments with it. Using a zinc finger would be another way to do it. The problem is that even if you could get it out of all of the T cells, what we know from research that we've discussed in some other episodes of TWiV is that HIV is probably lurking in an integrated form in other cells as well. So that even if you could clear it out of all of the T cells, it's likely to come back.
Yeah, that was
Right?
that
Fair
was
statement?
Kathy Collins, right?
Yeah, right.
Yeah.
Yeah, you have to figure out how to get those progenitors, those hematopoietic precursor cells.
So what you have to do is you have to combine that with the therapy that does the transplant back with cells that are resistant to HIV, like the CCR5 negative cells, something like that.
All right, Part B. This led me to wonder what happens to the original T-cell population of AIDS patients who go on antiretroviral therapy after their T-cell levels go down to dangerous levels. After the antivirals get the viral level down, what happens to the T-cells that are carrying the provirus? When they get activated and start producing virus, it seems like the medicine will prevent the virus reproducing, but will leave those cells around. Do the infected cells eventually get killed off by cytotoxic T-cells? Do AIDS patients who start taking antiretrovirals after their T-cell levels go down lose their previous immune memory and have to get revaccinated for things they were previously vaccinated against or exposed to? All right, now I sent this over to Kathy Collins this morning.
Oh, good.
And
Excellent.
she answered. Wow.
Good.
Okay.
Current evidence suggests that actively infected cells will most likely die after a couple of days from the toxic effects of the viral proteins and or the immune system. On the other hand, if viral gene expression is turned off, latent infection, then the cell will most likely live its natural lifespan, which could be years if it's a memory cell. These cells form reservoirs of provirus that can restart the infection if drugs are stopped. Then to the second part of that, what happens to previous immune memory? She writes, great question. Whether previously vaccinated HIV-infected adults, including those who receive routine immunizations in childhood, should undergo repeat vaccination after starting HAART in order to restore immunity is unclear. She doesn't. No one knows.
Wow.
Right.
So that's a great question, but we
That's
don't
terrific. Yeah, there were experiments done. I remember seeing this in an ASV meeting, maybe as long as 10 years ago, when the HAART therapy was in the early stages of development, but it was obviously working, where they asked whether you could take a person off the therapy and whether they were permanently cleared. And the answer was no. You take them off therapy and the virus comes back, the T cells get trashed.
That's right. All right, Part C. Finally, the whole discussion made me wonder where the T cells are replenished from. I've read some papers that say that as you get older, more and more of your naive T cells come from existing naive T cells in circulation, reproducing rather than going through the thymus. But for transplant and AIDS patients where the circulating naive T cell population is mostly gone, their bodies must somehow be changing that so that hematopoietic stem cells make immature T cells that go through the thymus, because otherwise they wouldn't get working T cells. But I could easily be missing something since this is far from my own field. I also asked.
Certainly doesn't sound like it.
I know. I also asked Kathy.
Oh, good.
I think the answer is that we really don't know, or at least I don't know. There is evidence that HIV-infected people have defects in their T-cell developmental capacity for a variety of reasons, and that this may contribute to low CD4 counts. All right.
Okay.
Well, I don't know the answer either, but that's a great question. Finally, I'm sending along a link to a paper on how T cells are produced at different ages and how this affects T cell receptor diversity. I'd love to hear it discussed sometime. The discussion on how aging affects the diversity of your T cell receptors seemed really interesting in terms of effects on stuff like effectiveness of vaccines in older people, and I bet there are a lot of implications of this stuff I don't know enough to even suspect. All right, we'll put it on our list for you of things to talk about.
What a great letter.
Thanks again for your wonderful podcast and for answering this computer scientist's amateur biology questions. Wow, pretty good for a computer scientist.
I'll say.
Damn
he is all over it. He's right on top of it. And you know what? We need to get an immunologist on the show sometime.
Here.
And we need to queue up a bunch of questions for the immunologist.
Yep,
right?
That's right.
Because I'm weak in that area, but I'm trying to learn.
All right, Rich, you are next.
Kimberly writes, "Hi, TWiV and Dr. Esteban." So here we have a listener writing to another listener.
That's right.
I did my graduate studies on baculovirus-induced apoptosis and consequently came across studies that you might be interested in for your course that was described in TWiV 145. So Kimberly is addressing Dr. Esteban, where he asked, what's his
course of viruses that affect behaviors, right?
Viruses that affect behavior.
That's where the zombie stuff began.
Yeah, that's right. That's right. Baculoviruses cause caterpillars to climb as high as possible. At late times in infection, the caterpillar melts, and I've seen this. They really melt. The virus just destroys them. And all of the virus-rich goo falls onto the lower leaves. These leaves are then eaten by new caterpillars, and they become infected. Terrific. Sorry, I can't remember what lab did the studies, but I believe it's described in an abstract from the ASV meeting that was at UC Davis in 2003, and I know it's referenced in the baculovirus section of Fields. Not a sexually transmitted virus, but I thought you might find it interesting nonetheless.
That's cool.
That's really pretty good.
Yeah.
Climb as high as possible.
And Kim is a postdoc in Diane Griffin's laboratory.
Could we get the gene for that and give it to people? Cool. The next one's from Gopal. Dear TWiV folks, I presume that someone would already have drawn your attention to this report that appeared recently in the British newspaper The Guardian. This is just in case no one has done so. It is a paper, it's an article called Chronic Fatigue Syndrome Researchers Face Death Threats from Militants.
Oh, this is so sick.
So these are stories of CFS researchers who've had death threats of various sorts. And Gopal continues, I can understand CFS sufferers being upset that the link with XMRV did not pan out, but it seems to me that death threats against researchers would make scientists more likely to avoid working on this problem, thereby retarding the search for the cause of this mysterious ailment and an effective way to treat it.
Exactly.
So Gopal, we've read his letters before. He is a correspondent, a science correspondent for the Hindu newspaper in India. Now, there is, in this article, they quote Simon Wesley. And in fact, on this science article we referenced earlier called False Positive, they have a picture of Dr. Wesley, who's at King's College in London, and they quote him as saying, people will rather go over the Niagara in a barrel than ever getting involved in CFS again. So there you go. You get death threats. This is what happens.
Right.
It's really
so
silly.
People have to understand they're actually poisoning the research by, not taking a rational approach to this.
So Myra McClure, a head of infectious diseases at Imperial College London, says, I published a study which these extremists did not like and was subjected to a staggering volley of abuse. One man wrote he was having pleasure imagining that he was watching me drown. He sent that every day for months, as an example. So I'm sure these individuals are not listening, but it's really nonsense. Let's do a couple more. Rich.
Al writes, Aloha, Vincent the gang. Al is in Hawaii. By the way, you guys understand that aloha has a huge meaning, right? That's more than just hello. That's like big love. Great stuff.
I think Al told us that once, yeah.
Yeah, maybe he did. Aloha, Vince and the gang. I notice you are going to talk about research funding in an upcoming episode. I think science research is too important to leave to the flaky Congress. Wikipedia is now a massive repository of human knowledge, and it is self-funding. I wonder if those who care could fund both targeted and untargeted basic science research through an independent science foundation where members donate, say, $500 a year, and a body of respected but somewhat out-of-the-box folks make funding decisions. I think it could work, and and this time of waning funding is a perfect time to start it. Love to hear your opinions. Best regards for the fine job you guys are doing on TWiV. Al from Hawaii.
Great idea.
Great idea.
Well, there are some NGO funders. I mean, the Gates Foundation, for instance.
Mm-hmm. Yeah, but he's talking about
Wellcome Trust.
People out there could give $500 to a specific foundation.
Right.
Understood.
And then we'd have a board to review grant proposals.
I wonder what Wiki's budget is.
I think they need a few $1 million to get going.
Should get this guy from Facebook to help. Just became the latest billionaire in the United States.
I'd like to start this, actually. I'm going to purchase some more spare time and do this.
Come on, let's go for it.
I think this. I don't want to make money from it. I think this is a great idea.
Well,
does Wiki also? Wiki doesn't advertise, do they?
No, they have fundraisers at different parts of the year. I think their budget is known. It's in the millions of dollars, and they try and raise it every year through fundraising.
yeah, that's right.
So you could do the same thing. I mean, in fact, Jim last time
ProMed does that.
Told us, what if you just got 1 million people to donate $10 each,
Sure,
and then use that to do science? I think this is a great idea.
Yeah.
it is.
Let's do it.
Okay.
I don't want to fund my own work. I want to fund new people going in the field who can't get NIH grants.
Right.
Good idea, Al. I could keep going on, but I think we have to wrap up.
Okay.
One more?
Sure.
From Neva. Hello, Vince et al. Your podcasts, including TWiP and TWiM, are extraordinary. Thanks so much for giving them to all of us. The article about academic publishing illuminates a source of frequent frustration for me, as I am blocked from viewing articles of interest. I know you are quite familiar with this issue, but this is a good summary, I think. Thanks again for your wonderful teaching. Neva is in Texas. So she sends an article. So when does academic publishing get disrupted? This was published in GigaOM, which is really a tech site. But it's all about not being able to get open access of journals and what can happen for it as a result. So it's a really good article.
Well,
Actually-
a lot of the articles that I've been trying to access, I can get the abstract on PubMed, but I can't get the full article without paying.
Yeah.
So
Neva is even in Buda, Texas. I actually know where Buda is.
Really?
Buda.
Because my
Wow.
daughter lives now in South Austin, but she used to live in Kyle, which is right around the corner from Buda.
Is it having problem with the fires?
Buda, I don't know how threatened they were, but they certainly need some rain.
Absolutely.
All right. Unfortunately, we have to stop email, which is too bad. We
Yeah.
could do another episode because we have as
Sure.
many more
There's-
left.
Yeah, there's
Okay.
a bunch.
Maybe we
This email is really fun.
Maybe we could do it next week.
If you send some to me too, Vince, I can read a few. I can relieve you of the burden of doing that sometimes.
I don't want to tell you that it's all in the Google Doc, which you get automatically each time. It's okay. I will email it to you
usually
next
sit in your
time.
office and I watch the screen.
That's right.
I understand.
No problem. We like your comments anyway. Rich, what do you have for a pick of the week?
I have this website that I just discovered. Maybe you're already familiar with it, you or Alan, called IO9.
No, I never saw it before.
I discovered this because of that hit about the computer gaming.
Right.
I don't know what it stands for, IO9. Okay? But it's a science website. Let me go down here to the About link. They have a little video about it, but it says it is a daily publication that covers science, science fiction, and the future. Okay? And it's just like a little, like a newspaper or something like that, with a whole variety of different science articles, including that one about the gamers solving the folding problem, and also stuff about science fiction, which I love. They have all sorts of takes on Batman and Star Trek and the rest of it. And so it's science oriented, but it's boundless. They have hardcore science articles and things of interest, and then a lot of science fiction stuff. So on the headline today is, under the topic of material science, how carnivorous plants could give us self-cleaning, self-repairing walls. Okay?
Cool.
So I think anytime I'm starving for a pick
You go
in
here.
the future, I'm just going to go here.
I
And
just
there's
learned
a wealth of them.
I just learned from this site that this morning at 5:05 the September equinox occurred.
There you go.
Do you know what that means, Dickson?
I sure do, Vince. It means that the day is exactly the same length as the night. How do you get to IL9? I'm typing it up, but
IO9, IO9.com.
I thought you said IL.
No, IO9.com.
Okay.
They got nice pictures of these picture planes.
Okay. He says we come from the future.
Okay, my pick is a blog, a specific post from a blog called Take as Directed. It's a blog written by David Kroll, who is a university professor who works in pharmacology and therapeutics. And he wrote a blog post called Trina Suderos on This Week in Virology. When do you fact check article content with sources? And he writes how Trina in TWiV 149 was discussing how she fact checks her stories. And there is a huge comment, 93 comments in this blog post from various writers, most of which say they don't fact check their sources. And included among them is Alan Dove.
Oh, he got a comment in there? Which comment is his? Is this down towards the bottom?
It's down towards the end.
Okay,
now,
because I read it when there were only 89 comments.
This has really disturbed me, but I'm not going to talk about it right now. I think we ought to discuss it next week when Alan is here, because I don't see why you can't check your facts. And a lot of these writers say we don't do it because of this and that and the other. And I talked to Trina, and she feels that there's just no reason. So check it out. It's a good discussion.
Yeah.
Maybe we could revisit it next week. What do you think, Rich?
Good idea.
All right, we have a listener pick of the week.
We
Sorry.
can do the whole episode over next week so that Alan can get his links in.
Yeah, we can do email and this. That'd be fine. I have no problem with that.
I have a pick.
I'm sorry, Dickson. What do you have?
That's okay. I have a pick. It's a movie that I just recently saw, which is one of the most mysterious and perhaps disturbing movies that I've ever seen. It's called The Tree of Life. It's with Brad Pitt and Sean Penn.
Oh, I saw that advertised this morning. It looked really good.
You got to go see it. You got to go see it. It's got a semi-religious ending, which is kind of cheesy, but it's got one of the most amazing 2001 Space Odyssey-like approaches to describing evolution. So you're asking if there's an evolutionary movie out there that you could watch to sort of bring you up to date. It doesn't have much explanation, but it has, I think, the best dinosaur scene I've ever seen. A velociraptor approaches a dying hadrosaur, and the hadrosaur is breathing, and you swear you're right in the scene. And I won't spoil it for you, but you should go see this, and you can hear it, and you can almost smell these two. You can almost smell the scene that you're in.
Cool.
And it's fabulous. It's just absolutely fabulous. But it's mysterious. It's quite mysterious. And it takes place in the 50s in Waco, Texas. So it's all about families and about the meaning of life and about the universe and about evolution and about life. It's too broad. It's too big, but I still loved it. So that's my pick of the week.
Dixon, have you seen Contagion?
No, I haven't, Vince. We've been threatening to go together, haven't we?
I know that Rich has. Do you know if Alan has seen it?
I do not know if Alan has seen it.
Because when we all see it, we have to talk about it.
Have you not yet seen it?
I have not.
No.
I went with a bunch of virologists. I went with Moyer and Musashi and McFadden, and we had a great time.
That's cool.
I'm sure it's good fun.
I've read several reviews of it, and one was in Science Magazine and one was in Chemical and Engineering News, and they both had almost identical takes on this movie. So I won't spoil it, but I'd rather see it first.
Actually, I let go a spoiler to my
Facebook.
my medical student virology class.
Okay,
because it came up and I said something. I won't say it again. Okay, and they all went, no, don't tell us. I blew it.
Exactly.
It's got a good cast: Brad Pitt and Sean Penn. I thought I saw on the review that Robin Wright was in it as well, but I don't see her listed on IMDb.
I don't know that, but I know that the kids that portray Sean Penn and other children in that family are just amazing actors. You swear they're not acting. It's amazing. It's a wonderful movie, but a little bit disturbing and totally mysterious.
Great. Sounds perfect.
Yeah.
All right, we have a listener pick of the week from Luis, who writes, Dear Twivers, first I want to congratulate you for the excellent podcast. I certainly enjoyed all episodes since last Christmas when a very good friend of mine, Alan Ryan, sent me the TWiV link of the show where he was invited to talk about XMRV. For me, it has been a great discovery and an excellent opportunity to broaden my knowledge on virology. My name is Luis, and I am a biochemist, molecular biologist working in Madrid, Spain, at the Centro de Biología Molecular Severo Ochoa, a Spanish National Research Council institute. My research is devoted to retroviral enzymes, particularly to HIV-1 reverse transcriptase, studying its fidelity of DNA synthesis and the mechanisms of antiretroviral drug resistance. I would like to propose a pick of the week, which I found particularly interesting for the quality of the seminars and talks provided. Perhaps it is not so much for the general public, but for advanced undergraduate students or predoctoral fellows. This is the NIH Videocasting and Podcasting website. By clicking at the Past Events section, users can access a pretty large number of talks, seminars, etc., some of them really interesting. A very good example of this kind is the seminar by Eric Lander, May 20th.
Oh, that guy's amazing.
On genomics. An impressive lecture.
Yeah, he's amazing.
Thank you very much again for your efforts to promote science in general and virology in particular, and long life to TWiV.
Hey.
Thank you, Luis. It's a cool site. I've been there. Yes. Do you have you on that site, Dickson?
I don't know.
I don't know how you get to be on this site.
Why would they have me there?
1,395 podcasts.
Yeah, pretty cool.
For the same reason we have you here.
Right. Can you search for Dickson? Can you search for Dickson?
I was about to, but
Stop it.
You guys can do it while I'm wrapping up here.
Everyone's been searching for Dickson, including Dickson.
That could be the name of a movie, Searching for Dickson.
That's why Desperately Searching for Susan. That was, by the way, starring Madonna, and it took place right here in Fort Lee, New Jersey. So,
yeah, they shot a sequence here at the Malcolm, the ballroom up here. I'm blocking on the name of it. The Audubon Ballroom, where Malcolm X was shot.
That's right.
There you go. Well, if you like TWiV, try TWIM and TWIP. You can find them at virology.ws. We have a fan page, facebook.com, This Week in Virology. Check it out. All these podcasts are at iTunes, at the Zune Marketplace, at microbeworld.org, and TWiV has its own special website, twiv.tv, where we have show notes and listener letters. If you are new to TWiV, you should subscribe on iTunes and leave a comment, because if you leave comments, it makes us stay on the front page of the medicine directory in iTunes, and that way more people can see us. And more people can learn about viruses, which is our goal here. Dickson Despommier is at verticalfarm.com, medicalecology.com, and Racaniello.org. Thank you, Dickson, for joining us today.
My pleasure. 150 shows. That's amazing.
Yeah.
Cool. That's a real landmark.
It is really nice. I'm really happy that we've lasted this long.
And there's no way I'm getting tired of it.
No, this is fun.
I'm really worked up here.
That's good. Good.
I could just do this forever. This is great.
Certainly enough material.
Yep.
Well, we'll keep going.
There you go.
Dickson, your sound was very good today.
Thanks. Using a high-quality instrument.
Rich Condit is at the University of Florida, Gainesville. Thank you, Rich.
Absolutely. My pleasure. What a gas.
All right.
Absolutely.
I'm Vincent Racaniello at virology.ws. You have been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.